Uh, good evening, good morning. Welcome everyone to ICANX, Connecting the World and the Universe. I'm your host, Professor Martin Thuo, all the way from the middle of the United States in Iowa. And our speakers for this month are going to be Maria Vicent, who will be talking today. And then there will be Mark Allen, followed by Chris Nyhouse and Zhuakai Wang from City University of Hong Kong. For today, we'll be joined by Professor Nicholas Peppers from the University of Texas, Austin, Professor Jianting Ding from Shanghai Jiao Tong University, and then our ex-challenger is going to be Longwei Gao. Our speaker, Professor Maria Vicent, comes to us from Prince Felipe Research Center in Spain. She received her PhD from University of Castellano and Berkeley, where she was working both in Spain and here in the US. After that, she spent some time with Professor Fauché at UC Berkeley. And then after that time, she shifted to more biomedical research, first at the Institute of Bioma, then at the Center for Polymer Therapeutics as a Marie Curie postdoctoral fellow. In 2004, she received the Marie Curie Reintegration Award. And at that time, she joined her current institution, Prince Felipe Research Center. And from then, she has risen through the ranks to head the Polymer Therapeutics Lab. She's the director at large for Control Release Society. She serves on several advisory boards. She's an editor of several journals and has received many awards, including the ERC Consolidated Grant, the Concepcio Alexandre Award, among others. She's a fellow of the American Institute for Medical and Biological Engineering. She's a fellow of the Control Release Society. She's very well published and holds several patents. And out of those innovations, some of them have been licensed and at least one has been spun out to a startup company. The resume is too long. I'm not going to take her time by reading more of what she is doing, but I'm going to hand it off to Dr. Maria Vicent. Maria?
Thank you so much, Martin, for a really kind introduction. And thank you so much for the invitation. And for me, it's really an honor to be giving this talk today. And I'm really hoping I can share some of my passion on the field of drug delivery and polypeptides today with all of you. So I should share my presentation already. So, as I was saying, thank you so much for the invitation and thank you for the kind introduction. The idea today is trying to explain how the importance of the interdisciplinary thinking, the rational thinking also, and taking into account that people who work on the field of biomedicine, trying to develop diagnostic and therapeutics, what we want is really trying to solve an unmet clinical need. And that's the purpose of my lab and many others. And we decided to work and to try to contribute to this challenging aim using polymer therapeutics, in particular polypeptide-based therapeutics, because we believe they have some advantages in front of other systems. So as I was saying, the motivation of the research in my lab is trying to understand which are the needs in the clinics and trying to understand also where with our technology we could contribute. There are many metastatic cancers, CNS disorders in this aging population where we are really getting worse and worse and we don't have any solution. And the attrition rates for the therapeutics are very low still. So deciding to really go with nanomedicine because I believe that trying to engineer the nanobio interfaces could give an improvement. we also have to really take into account that we are working with complex nanosystems. And for those, we have to control not only to understand the biology, but also when we implement the chemistry, we have to really control the characterization to know exactly what is in the vial. Because one of the things that we try to do always in my lab and many others is trying to design and to start doing chemistry and science from the very beginning, but thinking always that we have to transfer this to the patient. So we need to make something that is well characterized, simple enough, and cost effective to arrive to the clinic. So apart from the chemistry and the characterization, when we work with complex nanosystems, we need to really think that the pharmacokinetics, pharmacodynamics are completely different from small molecules. So we are working and trying to use and implement adequate preclinical models because this is so relevant. And we need to characterize them to understand if they are relevant for the questions we are asking, and also to understand and to correlate this preclinical clinical scenario, because at the end, as I mentioned, we should arrive to the patient, and the patient heterogeneity is huge. So in the whole process, also, if we are able to identify functional biomarkers for our nanomedicine, we'll be able to identify which patients or which population could benefit for the nanomedicine we are designing. So summarizing in this slide what I mentioned before, the approach we are taking is an iterative approach where many things are playing together in a convergent manner. So we are understanding the needs, trying to understand the biological barriers to cross, and to control this bio-nano interface with the chemistry and the tools, the chemical tools that we do have, is gonna help us to reach and to, again, facilitate the life of the patients. When we talk about chemistry and design, we are not only talking about identity of the compound, but also solution conformation, because not only the identity of the nanosystem, but also the solution conformation is the one who is going to rule all the trafficking at the whole organism level, at the cellular level also. And always keeping, as I mentioned, translation in mind, depending on the type of diseases you are targeting, you have to also think and to make your decisions from the very beginning. If you need to go for biodegradable systems because you are using chronic application like Alzheimer's, neurodegeneration, these biodegradable systems should be well-defined, well-controlled, and you should develop tools to characterize those systems and how they behave or the fate of these systems, not only ex vivo, but also in the physiological fluid, because you will need that for validating methods when you are arriving to the clinical scenario. And also to this translation from preclinical to clinical, you also have to really select those models, adequate models, and also be sure that the chemistry implemented in the process are able to be scaled up. So we are keeping all this in mind in our systems. So what we do is working with the chemistry and the physics to have all this well controlled in an iterative manner, also characterizing the in vitro models, not only for which cells are we working, also trying to mimic in these 3D spheroids, organoids, every time more complex type of systems, to identify those endogenous triggers that, as you will see later in my talk, we are using as triggers for activating our nanosystems, the endocytic rates, or any markers that we need to follow. Also go into the in vivo. You're talking about tumors, for example, or inflammation, is important, the vascularization for the nanosystems. So every model that we are working on, we try to characterize regarding also endogenous triggers, some specific biomarkers, and also vascularization, a specific type of predictors or biomarkers that help us to accelerate the process from the design to the real activity. And implementing this, we are working in different lines of research, but I can say that just modifying the polymer per se, because we are working with polypeptides that are kind of protein type structures. It's versatile, you can modify, charge, size, shape. We can target specific organelles, cross specific biological barriers, or go to specific organs, so we can really target or modulate the fate of the systems. We are implementing techniques for all our projects that involves imaging as well as combination therapy, because we are strong believers that all the pathologies are very complex, and if you are not tackle things in more than one site, the body is too intelligent and it will be able to escape from what we are giving them. So we are really implementing combination, not only combination of drugs, but also combination of therapies. We have examples in a spinal cord injury, for example, where we combine cell therapy and nanomedicine. We are combining radiotherapy, for example, with a combination of drugs on prostate cancer. So this is really benefiting for the therapeutic approach that we are looking at. We are working a lot with the immune system. We are working with very nice collaborators in Tel Aviv and Portugal in immune therapy, but also trying to understand not only making vaccines, but also understanding tumor microenvironment. The immunotherapy has a major role there. And we understand that anything that we do, especially in cancer, for example, it has to do, the immune system has a major role in all the diseases that we are playing with. And all the whole time we are looking for these functional biomarkers. Currently in my lab, we are focusing, the more active projects are in metastasis, is breast, prostate, melanoma, pancreas metastasis mainly, and also in neurodegenerative diseases. And we have started in the recent years, in the last couple of years, to get interest on pediatric diseases because of our tools for CNS delivery, intranasal tools maybe that we are looking at, like pediatric glioblastoma, and also we are looking at different type of rare diseases that are important in the pediatrics, because I believe this is really a very important family or population that we really need to have. And how we do all this? So within the field of nanomedicines that is very broad, and there are many different designs, we decided to work on polymer therapeutics. So it's a polymer-based nanomedicines that the field was first coined by Professor Ruth Duncan. When I was very lucky to do a postdoc with her, and she's been my mentor for many years, and continue to be there for this big field of polymer therapeutics that is evolving very quickly, and really is going towards a hybridization, as I will see. But still, the concepts and the design features are those that Helmut Ringstorff also, in the 70s, designed it as polymer drug conjugates, for example, is the one that mostly we work with in my lab, because the work I'm going to show you today is mainly focused on small molecules. So when we are designing a polymer drug conjugate, there are different features that you can modulate to fine-tune all the things that I mentioned before, all these bio-nano interfaces at the whole body distribution, and also how things are really going inside the cell. So these are the polymer carrier. These are the drugs that they are conjugated, and mainly, as I mentioned, we will look at combination therapies, how these drugs are linked together. And the linking chemistry in our designs are a critical feature and differential from other type of nanomedicines like liposomal formulation or nanoparticles, because usually we don't encapsulate, we conjugate. And also, depending on which biological barriers you need to target, we need also active moieties. And I will show you, I will try to drive you through the rational design we are implementing in each of these features, and you will see the versatility, and I'm hoping for this. Starting to talk about the polymer, the background, the importance of selecting adequately, we decided to start working with polypeptides because they are biodegradable, and because the aim of many of the pathologies that we are targeting are for chronic administration. We believe using polypeptides or using biodegradable materials can help you to secure this therapeutic window of safety that you are looking for when you are designing a nanosystem. Polypeptides are also very versatile materials. Apart from biodegradable, they are degraded by endogenous metabolites, and these endogenous metabolites they are the amino acids. So there are many amino acids, natural and unnatural. So the versatility is huge, and you can really play. And there are many examples already in the clinic, like Copaxone is a polymeric drug that is used for multiple sclerosis, and it's a polymeric drug, it's a polypeptide. Or Star Pharma has this dendrimer with polylysine that is a viruside, for example, for topical administration. But more and more, as I was mentioning before, things in the field of polymer therapeutics and in the polypeptides in general is getting more complex and going towards more hybrid materials and combination approaches, because we are trying to mimic and to engineer a thing that resembles the nature. And for that, polypeptides are great because of this functionality and the conformation. At the end, they are behaving as proteins. So they have the beauty of this versatility, but at the same time, they are quite challenging materials, because to control the conformation, that is a key feature. Also, you have to really understand what you have in hand. But they are really have the benefit in the clinic, demonstrated that they are good, and they are really capable materials to do many things. But I mentioned, so we need to first control them. We decided to work with polypeptides synthesized using ring opening polymerization on N-carboxy anhydride. So this polymerization was described in the 50s. So when we started working with polypeptides, we thought they were going to be easy. But when we really look at the reaction, and you see here how many side reactions you can get in the ring opening polymerization of NCAs, we realized it was not trivial. And because if you want to control the polymer carrier, and it's something very important if you want to really push this translation, controlling the manufacture, bias-to-bias reproducibility, you need to really go to the normal amine mechanism, where you have initiation, that is non-nucleophilic initiation, and to have a living polymerization, and to avoid this side reaction. We were playing with the system a couple of years until we arrived to the right way to have conditions to not only get the materials that we wanted, but also in the amount, in the length, and in the concentration that we wanted at the end. So in this polymerization, and this very demanding one, you need to have very pure NCA materials, and you have to have very highly controlled the initiation process, because the normal amino mechanism is the one that is going to give you a very low polydispersity. But if you are using the basic mechanism of initiation, then polydispersity will increase. There are many people working on that, very brilliant people in the polypeptide field, like SLAD. There are many pioneers in the field with different methodologies, so we learn a lot from them. And helping from this SLAD method that it was reported with this source to avoid, to diminish this basic character and to control initiation, we end up with a process where we use BF4 salts and non-nucleophilic initiators, where we could control how we started the ring opening polymerization of our polypeptides and keep them as a living polymerization, so we can control really the N-terminus, what it was very important, the length, because one of the limitations with the methodology so far, it was that you could arrive to certain degree of polymerization, but very large polypeptides was difficult to get because they collapse. So with this polymerization, even if it is very slow, but you can reach large molecules, sometimes you need them, sometimes not, but it is kind of versatile. And thanks for this, we were able to start really producing bias-to-bias reproducibility, playing with the architecture. And this was the origin of the spin-out company that Martin mentioned in my introduction. And this company now is grown very, very quickly, and it's a CRO, CMO. They are making these polypeptides in large scale, what is also helping our science, because it really supports that the materials we are using and the system that we are synthesizing in the lab, at a certain point, we will be able to scale up and to manufacture to arrive to the patient. So the whole message for this polymerization, what we learned in the process that it was not short, is that you need a high quality monomer, NCA monomer, a precise control of the polymerization. And this will allow you an accurate control on the polyamino acid at the end, not only linear, but also any type of architectures. And you can follow and reach GMP characteristics with the ICH guidelines. So this is possible to be done, but you have to really control the process. So this was achieved by the company by supervised reproducibility and large scale production. And also what we observe in the process of the different architectures is that we can really play and try to modulate the use of polypeptides for different applications because they behave differently in the body. What I mean with that, so if you use the linear polymer or you use an star-shaped polymer, for example, the final conformation is very different because the aggregation behavior and the behavior of the star-shaped polymers in the physiological fluids is very different from the linear one. The capacity of the loading is very different. So these star-shaped polymers that are not pure dendrimers, that they are grown from a branch core, allow us to really go or move from a small amount of nanometers that we got with the linear polymers, increasing the concentration. We could reach quite a large particles without really going to use a large molecular weight polymers, for example. At the beginning, we didn't understand the behavior because what we saw, we haven't seen it before with the linear. And as you see here in the picture, the tomography picture, we started working with a BTA core. We thought that the BTA had something to do with the process of aggregation because it was aromatic. And the first one that we synthesized had a 50 amino acid unit. They were polyglutamates. They were a polyanion. And we observed that they were kind of particles of around 100 nanometers of radius. And they were very soft, no structure inside the structure, not ordered structure. So we didn't have any order due to the aromaticity of the core. However, with increasing the concentration, we increase the size and we decrease the zeta potential. And also another feature that we saw at the time is that this structure, these aggregates, they were able to be disrupted very quickly with ionic strength. With salts that were non-ionic, they were not disrupted. And also that the only shift in the circular dichromosome was due with the stars and it was due with the branches. So we understood with these first comparisons with our first star polymer that the length and the polymer, the polyanionic character of the branches had to be the major role there in the aggregation behavior. We were lucky at that time because we went through a paper that the physicists were publishing, Muthu Kumar, where they was describing a theoretical concept called the ordinary to extraordinary transition behavior. And it's really based on what we observe, because at the beginning it seems counterintuitive that when you have a lot of polyanions, they don't want to be together, but they do, because the huge amount, the counteranion cloud surrounded by the polyelectrolyte chain, that is because at the end you know that polyglutamates are COI minus sodium positive, so they are sodium salts to make it soluble in water. This cloud of anions plus the counteranions generate transient dipoles that are very strong, and they are doing it very quickly. So these transient dipoles really facilitates the order or the structure. So if you are able to modulate your starting unimer, you could modulate also what you have at the end on your final system. And this is important because if you remember, the solution conformation is key for controlling the PK/PD in a physiological environment. So with this in mind, a more systematic work was done with Alex in the lab, where we modulated and we tried to understand where it was the inflection point, and if with these versatile carriers we could really go from these collapsed nanoparticles to more fibers or different architectures. So the hydrophobic hydrophilic ratio between the core and the branches, also having different type of amino acids in the core, it was explored to see if we could get this differential aggregation behavior. And we did. Alex managed to see very nice fibers when we have very short branches So with a very hydrophobic core and very short branches, the supramolecular assembly is the one who is the major role. We don't have here the extraordinary ordinary behavior. We have a supramolecular assembly. When we shorten the branches and to have around 25 units of glutamic acid, so we have discrete nanorods. And then when we go up to 50 or more, then we have the original example that I showed you. So just modulating the length with the same core or increasing hydrophobicity of the core and shortening or making more important the amount of moments in the branch, you can easily go from fibers to particles. And not only that, you can modulate the different behavior or the different amino acids in the core. So Alex managed to have a huge family of different architectures that you can really think of different applications with all of them, because we will have a huge impact on the PK/PD depending on where you're applying them. Not only the length of the branches, but also the modification of those branches. So for example, Fernanda is working with Ima and Aroa. They are working on making more type of a nanogel because they are looking for an intranasal application. And what they observe with this system is that depending on the degree, for example, of tiols that they are adding to the branches, they can go from nano to the micro scale. So not only the length of the branches, but also how these branches are modified. It could help you to modify the size and the shape of the molecules. But if you remember, when I mentioned at the beginning that these molecules or this aggregation behavior, if you put them in ionic fluids or ionic salt, it completely disrupted. It reverses equilibrium. So we need to stabilize them. And we are using this bottom-up approach to organize and to design our systems. So we really modify with orthogonal functionalities our unimers. Depending on the design of the unimers, the size of the branches and the hydrophobicity of the core, we have different shapes and sizes. We can even put the drug inside this unimer depending on how accessible we want the drug to make it. And then once we have the preform form, the size, we capture them with crosslink, either with biodegradable or non-biodegradable systems, depending on the application. And this, once stabilized, you can also decorate the surface with different things. You can decorate the surface of the particle with targeting moieties, with molecular probes, imaging, antigens. So if you want to hide something that is very exposed, you will conjugate it or you will derivatize the surface of the system after cross-linking. If you want something that be hidden and be controlled mode release, you will put them in the core of the system. And once you click, this is an example that when the orthogonal functionalities were an alkyne and a site where we did click chemistry, and you increase the concentration of the ionic salt or the concentration or the presence of the ionic salts, you see that the size were completely stable. So we have really stabilized the system. And these systems are stable, even this cross-link material, for more than 4 days. So we did this vast bioreproducibility in the lab at a bulk scale, and we managed to have very nice results up to 100-gram scale. And we now wanted to transfer to a scale up. But one of and you have to be very critical also with yours. So these materials are beautiful and we're working very nicely in many things, but to go to a scale up and to go to manufacture, as they have to have a concentration for the self-assemble, one of the key features is this CAC, this concentration, this aggregation, the concentration they need to do this assembly. And to keep this reproducible, you need to implement flow chemistry. So in the lab, to do the first batches at small scale, you don't need this, but to go to a scale up, you need to implement microfluidics and flow chemistry. So this analysis of the critical attributes are very important also to move forward. So summarizing, with this type of materials, you can have unimers where you conjugate a drug. With the unimers, you can also have combination of drugs. You can cross-link them, and afterwards or before, you can decorate the surface depending on the application. So that's the thing. And why is so important to control just the carrier alone? Because of the different PKPD, as I mentioned many times already. As you see here, this is a biodistribution, a quantitative biodistribution that we have done. We compare polyglutamate, linear polyglutamate, star-shaped polyglutamate, and the cross-linked polyglutamate of this material. So they have 50 units of the branches, 50 units, and this is the cross-linked material. So you see that the cross-linked material has around 4 days. The linear one, it has less than an hour. So it really affects the half-life in blood. The time these systems are circulating in the blood is completely different. Not only that, there was really a surprise for us the first time we saw this biodistribution. The first thing, because these are polyanions, so we expected a renal excretion because this is known for the polyanions to be excreted in the kidney, but the cross-linked material with the -60 zeta potential as a total charge, you see that they are really going through the kidney. But what it was striking for us, it was the accumulation on the lymph nodes for the cross-linked material. That was 40% of injected dose and after intravenous administration. And it was the first time we saw these differences. This opened up in the lab a lot of projects in the nanovaccine, not only nanovaccine, also in the metastasis in the lymph nodes. And we are really working hard with Elena. And I hope in the next time I'm playing to you, because this work is not published yet, I will be able to show, because it's a beautiful project that we are running with Elena, and also now in pancreas with Ronnie Sachifainaro. And they are really nicely working with a special mechanism of action. But looking at this tropism, not only going to the lymph node has importance because of this renal accumulation, we could really design very nice projects with collaborators from the hospitals where, for example, this first example is showing an application in an acute kidney inflammation. So in this case, with Adrián Ramos from La Fundación Jiménez Díaz in Madrid, they came to us saying that the curcumin was an anti-inflammatory and could be a very nice drug to go and to solve or to prevent kidney inflammation in this situation. So we conjugated the curcumin after the cross-linking, because one of the things is clear here, the curcumin couldn't be conjugated before cross-linking because it was not stable. So we need to do it afterwards. So we conjugated, and because of this nice renal accumulation, what we found is that the conjugated material, it was very nicely decreasing the levels of urea and creatinine in the model. They induce the inflammation with folic acid. And not only the creatinine and the urea, but also they look to the kidneys and they have a completely reduction of the oxidative pathways. And you see you have much less caspase-3, the tunnel was much more diminished, and also oxidative pathways were diminished. So we enhanced significantly, because in here, what we did is like we accumulate, we have a tropism to the kidney, and the curcumin was linked to the polymer through an ester bond, and the inflammation, it has acidic pH surrounding it. So the release kinetics of the curcumin makes the polymer to act as a depot at the same time that they were degrading. So the curcumin was staying much longer time in the site of action. So that's why we have a so enhanced activity in the preventing of the cell death in the kidney in vivo, and also in the oxidative mechanisms. The same concept was applied a few years ago, before with using an APAF1 inhibitor. This was my first, I wanted to put it because this was my first project when I first arrived to Valencia. I arrived to a group of Quique Pérez Payá that is now still working with Mar Orzaez, that is the girl who take over that his group. And it was a very nice project because it was looking for drugs that they were interfering in the protein-protein interactions and the formation of the apoptoson, looking for anti-apoptotic applications. So that was a very successful approach, because the possibility for the polyanions to accumulate in the kidneys in an ischemia reperfusion swine model, for example, where they induce ischemia just hitting the animal, hitting for 90 minutes, you see that they are very different morphology in the ischemic kidney. So you see that after the treatment with our QM50C, that is the name of the conjugate at that time, you could reduce completely very nicely the apoptosis, and also the caspase-3 activation. It was inhibition, sorry. It was almost complete. This conjugate was transferred or licensed to a company, and they are applying, not in kidney now, but they are applying a derivative of this system for problems with the ears, for some apoptotic death in the ears. So you see that they're controlling the polymer and understanding the tropism that you can get just with the carrier and modulating a conformation give very nice results. The second feature I mentioned at the beginning is the linking chemistry. I mentioned with the curcumin, for example, or with the APAF inhibitor. In the APAF inhibitor, it was a protease labile. I didn't mention a protease labile link chemistry. So you need to design the linkers to achieve this control release. If you are looking for therapy with small molecules, you need this release. If you are looking for dyes or for probes or imaging, maybe you don't need to release, and then you can use click chemistry, and it's a non-biodegradable system. So understanding which linking chemistry you can put is also important to control not only the fate that you do with the polymer, but also the PK and how fast or how slow will your drug release. We like a lot now lately the self-immolative polymers, linkers, where we use the redox behavior. And we are using this because in many pathological sites, especially those in the CNS disorders, has a strong increase in the GSH. So we use the GSH enhancement to trigger the release of the drugs through a self-immolative manner. But it's very clean and very site-specific. In the case of the linkers, I will put you a couple of examples. One for diagnosis, where the linking chemistry we use here, it was not important regarding degradation, because what we wanted, it was really have a good signal. So we use here click chemistry, that is a non-biodegradable linker. But keep in mind always that the polymer we are using is biodegradable. So at the end, the whole material will biodegrade in endogenous amino acids. So the first application, it was through a European proposal where we collaborated with many people. And the idea here was using FRET probes, and the FRET probes for like a substrate or protease specific. So it was, and the role of the polymer here, our role here, it was to have this enhancement of the tropism. So taking these protease specific probes and drive them to specific areas of the body. The one that worked the best with the polyglutamates, we explored several, it was metalloprotein 13, and we application on osteoarthritis, because the metalloprotein 13 is overexpressed, is 13, sorry, it was up, is overexpressed when you have a damage in the osteoarthritis. So we use different polymers, derivatization with different linking chemistry, the spacers, and as a substrate, we use a FRET probe that was designed by a group in France, by Vicent in the CEA. So they use a protease labile specific substrate, and you see here two dyes, that is the FRET pair. So the polymer, we optimized the length of the linker, and it was a non-biodegradable linking chemistry here, but just the adequate length and exposure of the substrate makes that even the specificity after conjugation increase a little bit, and also the distribution also changes because of the polymer. So we didn't diminish the specificity of the substrate towards the metalloprotease 13 when compared to the others, but we increase even a little bit more. What we managed to do it even more is that we have this homing device. The polyglutamates are polyanions, and we understood that the macrophages kind of like them a little bit. So we believe that they were an active role, and they accumulated because the inflammation was not really huge in this in vivo model, because the damage they did in this osteoarthritic model was very little inflammatory. But even the little inflammatory side, you know that because of the APR, the nanomedicine could accumulate there. And because of this accumulation, and you see here, we have a much nicer signal for a longer time compared to the free probe. So we could have a signal even earlier, so we can accumulate the probe earlier to have a beta signal. And this is in the model. You see here, it was not only 8 weeks, as it was for the free drug. We already at 6 weeks, we have a significant difference. And when we put this in the perspective, not only in the diagnostic part, but also in the therapeutic bit, we first demonstrated that it was protease specific. So we use as inhibitor provided by Sanofi, that it was a specific inhibitor for metalloprotease 13. And you see that with the inhibitor, there was no signal in the model and it was completely diminished. And this inhibitor was also a therapeutic. And in the model, which we could see after the treatment, that the disease or the pathological or the injury that we induced, it was recovered, as you can see here and quantify in this graph. So in rationalizing and making the linker adequate, we could really hone our protease and enhance the properties of this molecule as a molecular diagnostic. But when we are talking about therapy, and especially when we talk about combination therapy, the linker should be releasable, should be degradable. So, and we want this combination because we are tackling more than one path at once, and this heterogeneity is very high. There is one example, as I put here, already approved. The Combiplex it was the blockbuster. It was the first approved in the clinics, demonstrating that combination therapy is the way to go because it really makes a difference, delivering to the same cell the combination of drugs in the adequate ratio that can make synergistic effect in cancer and in other diseases. We are not only encapsulating both. What we are doing also is if both targets that we are looking at are in the same cell, in the same site, so we are putting both drugs in the same polymer and fine-tuning the linking chemistry to elaborate the release of one drug in front of the other. This is also quite challenging regarding the characterization, but that's something that is part of the game, and we need to really be aware that we need to characterize. So we put this in value in a project in breast cancer, for example, where we were combining drugs that they are already in the clinics and drugs that they were in particular mechanisms that we wanted to stop in the metastasis. So we were looking at tumor-derived exosome release as a possible mechanism to stop metastasis progression. So the idea was to select the drug and to select one from those in the clinics in a subtype specific manner, because we were working in breast cancer in this project, and combining with a drug that has a mechanism that can stop this tumor-derived exosome either release or biogenesis. So we did a screening approach to look for those combinations. The idea was trying to find these combinations for each specific subtype, and then with this combination, conjugate to the adequate polymer with the adequate linking chemistry. We have to do this also in adequate models, as I mentioned before. So this in breast cancer, you have the in vitro and the in vivo. The in vitro for the screening, we have the 2D, and we have also spheroids and organoids 3D that are more reliable models or more similar to those that we encounter in the animals. And also with the animals, we have many different. So it's very important to select those models, as I mentioned at the beginning. So for example, for breast cancer, we did these screenings in spheroids with a different subtypes with TD from commercial derived cell lines. So each of them, they were well characterized, and you need to have techniques to identify and to stabilize them. But also we have organoids derived from patients, and we use a process described by Hans Kleber where we use a matrix. And in the difference between the spheroids and the organoids are trying to keep the heterogeneity of the original tumor. So we have, in collaboration with the hospital, we managed to get like a small biobank of different organoids where we are using them for the different specific type of approaches. And the behavior of the nanosystems in 3D versus the 2D are completely different. They behave very different. So we have to be very careful. To do a first screening, do it in organoids is not possible because it's too expensive. We don't have enough material. So we do the first screening in 2D, validating spheroids, and then we validated the final conjugates in the organoids derived from patient apart from the in vivo. And from the in vivo, in the animal models, we have selected two different ones. So we have the 41 model that is immunocompetent because it's a very aggressive, and anything related to the immune system, it could be answered using this model. And 231 model, both of them triple negative because it's the type of tumor that it has is less stratified, or it's more heterogeneous than others. And as you see here, we fully characterize both models. So we know exactly which day the metastasis started. Both of them are orthotopic and both of them are metastatic. So the metastasize in the lungs and in the lymph node. And the 41, for example, started already in day 3. The 231 started in day 28 to metastasize. And we even identify a biomarker, the growth factor stimulating factor in the 231 that tells us when this shift in the metastasis was coming to place. So using all these models, we can select the drugs. We use this screening with the bliss ratios to select the drugs with the different, with those that has a synergism. And also for the exosomes that are very important, as I mentioned, we did a screening, not only with cell viability, but also to understand which of those drugs or combination we are able to stop this release or to stop the biogenesis of the exosomes. And for that, Zoraida with Esther and David, they implemented also a type of a high throughput approach with alpha screen technology, where we identify when those materials or which drugs were really able to stop the release or modifying the biogenesis of the exosome. So using alpha screen technology or in-cell by flow cytometry, we could really, with this high throughput screening approaches, either selecting the drugs and be ready for those to be conjugated to the nanosystem. For the linkers, once we have the drugs should have adequate functionality to be conjugated to the polymer. And if they are small drugs, usually what you are waiting for is that they are internalized in the cells through endocytosis. And then once arrive to the lysosome, the pH or the presence of different proteases will cleave the polymer and the linker and release the drug, because they are small molecules and the ones that I'm showing you here. This lysosomotropic delivery was reported by Dubé and Helmut many years ago, used a lot by Rutankan, Kopechek, and many others, with many examples in the field. And it's really working very nicely for small molecules and also for enzyme replacement therapies. So it's the one we are really using in the lab for small molecules. So we selected a couple of drugs with all the screening process. In this example, we've had an aromatase inhibitor and a chemotherapeutic agent, and we conjugated both to a polymer, to a polyglutamate. Both of them, we can either conjugate through a protease-labile linker or through a pH-labile linker. As I mentioned, because they are the triggers in the cells, but also thinking that the pH is a trigger that could be also in the tumor microenvironment. So we are looking for a faster release and having an effect not in the intracellularly, but also in the tumor microenvironment. So these linkers were the protease-labile for cathepsin B, and then for the pH, we go also for the accessibility and looking for a faster release or a more controlled, more slower release with a shorter hydrazone or a longer hydrazone with the EMCH linker. So you see the first design with the protease-labile, we demonstrated that having both drugs in the same polymer have a benefit in front of the others. And also the ratio between the chemotherapeutic agent and the small molecule has an effect. And this effect, it was working in this case with the high loading versus the low loading was working better. And you see here, this is stomach volume. Always we do the same controls. We have a great accumulation with the conjugate compared to the free molecule, a greater half-life, and a different biodistribution. In this case, we use the 41 model. And as we were using doxorubicin as the chemotherapeutic agent, cardiotoxicity is a problem with doxorubicin. As you see here, there was no accumulation in the heart anymore after conjugation, what is typical for the polymer drug conjugates of doxorubicin, being reported also in those that transfer the clinic. When we change to the pH labile, the chemistry, we always try to control as much as possible the chemistry with well characterization and so on, with a smaller or longer linker, with different loading chemistry. We found that also both of them were very active, even more active than the protease labile. As you remember, it was around 30% diminution of the tumor model in the 41 model. But, and all of them were very safe regarding cardiotoxicity, the longer and the shorter linker. However, the longer linker, we found a little bit of hepatotoxicity. We have a little bit of inflammation in the liver. So, and that was because it was a very too fast release of the doxorubicin. So we decided to move forward with a shorter linker, with a hydrazone shorter linker. And you see here that both drugs, both ratios, high loading or low loading, both were very similar. It has very similar effect. No, you see that the loading of the drug is also important. You see the red has a much stronger effect than the brown. And the difference between these two is that the loading of the doxorubicin is much lower here than here, you see? So with the red one, that is low loading of doxorubicin, higher loading of the endocrine agent, we progress and we look at the metastasis progression, because that's something that we wanted to see also. And this pH labile release is allowing us to be more effective in the metastasis, because at the tumor microenvironment, we are already stopping this metastasis progression, this exosome release, for example. And we saw a very nice effect on the lung metastasis that we have in the 41 model, very nice effect. Analyzing transcriptomics, for example, is telling us that the immune system, it has a major role, and it has the more effective, it really activates also the immune system. So we are really acting on many responders at the tumor microenvironments, and we are really diving into this effect. So really designing the polymer, not only polymer, the drugs, and also the linking chemistry is also making a major role in the therapeutic output. So just to finalize, my final example is this active targeting. It's the gold standard. So if we could actively target every single place, it will be very nice. But there are not many targets that are known that are very differential from a normal tissue, from an active or a pathological tissue. But there are others that you can use, like the HER2, and there are many examples. And in the case I'm showing you, for example, to really bypass the blood-brain barrier, if you are with nanomedicine, if you are not working with active targeting, it's very hard to bypass. So for us, the active targeting was very useful for the CNS disorders. And we could really see an effect in different applications, in brain metastasis for breast, Alzheimer's, and I'm showing you a little bit of both. But in the brain metastasis is an unfinished work. Just show you the distribution. So the blood-brain barrier is a huge barrier, and it's one of the more challenging ones to cross. And we are also exploring the nose to brain barrier with intranasal delivery, because I think is some applications, because they are less invasive, and for some chronic applications could be very nice. So we are really going deeply in that direction. But the first example that I'm going to show, and the validation of our system with it, was IV. So the blood-brain barrier is a very strong, tight material, and you need really an active mechanism to go through. So we are using transcytosis, receptor-mediated transcytosis, to really be able to transport from the blood to the brain side, our cargo. So to do that, there are many linkers that has been reported, but very few that work properly. So we believe that we try, and there are ours and others, like Pepe Battaglia, for example, has a lot of nice work with LRP1 receptor, and Barrett was the original with describing angiopeptin-2 as the peptide to target LRP1, angiopeptin-2, that is in the clinics conjugated to paclitaxel, for example, in phase 2. But this LRP1 receptor is overexpressed in the blood-brain barrier when you have a metastasis from breast, for example, as you can see here, and also in the early stages of Alzheimer's, not in the late stages, but in the earlier stages when you're looking for a neuroprotection, or is really also overexpressed the LRP1 receptor. So we decided to work with that and derivatize in our systems with this 19 amino acid peptide to force on to look for this active targeting transcytosis mechanism. So the first thing was we conjugated to the polymer. The only one that worked for us and managed to bypass the BBB was the cross-link system, because it's the one that it was longer circulating in the blood, so has more time to expose the targeting to the receptor. So we had around 1.5% of the injected dose going to the brain. It's not marvelous, but it's more than nothing. And we really managed to see differences. In a really nice collaboration with Manuel Valiente, he has a model in CENIO in Madrid. He has a model of brain metastasis from triple negative breast cancer cells. So you see the GFP label 231 cells, and the red blood vessel, because our polymer is labeled with cyan 5.5. So with this cyan 5.5, it can be able to extravasate and to colocalize with the tumor cells. And Fernanda is really now having very nice results diminishing this metastasis in the brain. And I'm sure shortly we will be able to show this data. But this was really an encouraging data to us that prompt us to move towards other applications, because as you see, not only this system colocalized in the cancer cells, but also going somewhere else. In this somewhere else is because our system, after intravenous administration, it really stays in the blood vessel for a longer time. So it really stains the vessel in the brain. So this is after intravenous administration, just after 30 minutes, you see a very nice labeling of the blood vessels. These blood vessels, they are not only those close to but also they are the cells close to the blood vessel are taking out the material, but also those distant from the blood vessels. And they are going to astrocytes, to glia, and to neurons. And they are nicely colocalizing with the lysosome. So they are internalized, the material is internalized by the cell. So we have a kind of a depot type of system that sticks in the vessels. So they are there for a longer time to feed drugs that maybe by themselves can go into the brain, but also by itself it could be transcytosed and then go to the distant places in the brain and be internalized. And this is very interesting, the distribution, because the more vascularized areas in the areas of interest of neuroregeneration. The olfactory valve, for example, the hippocampus are really well vascularized, and this is really helping us for applying this system in neuroregeneration. So with that purpose, we look for drugs that target the anti-inflammatory, antioxidant pathway, looking for treatments for neuroinflammation, Alzheimer's, neurodegenerative disorders. And that's really the purpose of this screening. So we were looking for drugs synergizing that were with the capability to have both actions, antioxidant, anti-inflammatory, that are the major triggers of neurodegeneration, but the very early ones, because we want to tackle at the very beginning. So we identify those combinations, and then we conjugate those combinations in our system and derivatize with the active moiety to look in an Alzheimer's model. So this combination therapy in this specific case was a propagule group that is common for these MAO inhibitors that are in the clinics. And again, was the curcumin as an anti-inflammatory drug. So we conjugated both. We had to do it afterwards because the curcumin was not stable before cross-linking. So we conjugated the material, the curcumin afterwards. And the propagule groups were done before, because it was an overexpression of the material in the unimer. And then we conjugate in the antiope through a bioresponsive linker. So we have the antiope with a cysteine, terminal cysteine, and derivatize the cysteine with PD, on the SATA modification, and we conjugate it through a bisulfite bond. So the amino acid, the 19 amino acids are linked to the polymer through a bisulfite linker. So with a hippocampus, with a ex vivo model, it's an orthotopic model. So the first thing we tried to understand, it was if our system was able to prevent a threat. So in this case, a threat, it was the A-beta amyloid. The A-beta amyloid would trigger inflammation and neurotoxicity, and the presence of the conjugate, it was able to diminish this inflammatory threat. The polymer was not doing anything. There was not an inflammation. So it was in that case, it was really nice and really safe. But what we also saw is that the polymer itself was doing something, because the polymer by itself, it was disrupting, as you see here, with it was able to neuroprotect, but it was able to trigger the amount of dendrites. So what we believe it is that the polymer, because of the polyanionic nature, is able to interfere with the fibrils, and these fibrils are disrupted, are not able to damage as much the tissue or the brain. So this interaction we calculated through NMR, and we demonstrated by electron microscopy as well as by NMR that the polymer itself and the conjugate were able to disrupt the A-beta amyloid. So we will not only have here the effect of the drugs, but we also have here the effect of the carrier. So we have also a synergism here. And when we move to the animal model, again, in the Alzheimer's, there is no good model for Alzheimer's, but in this ARCA-beta model, we again have a similar distribution as we show in the BALC animals. It was safe, and after so many injections, intravenous injections, and it was accumulation in the hippocampus, for example, as well as in the cortex. And with this combination of drugs, it was very nice to see that in comparison with the free molecules, the biscurcumino, we also validated with genistein that is also in the clinical trials for Alzheimer's disease, that the conjugation with our carrier, it was able to recover, for example, the olfactory memory of the mice. And the olfactory memory, is one of the things that the patient from Alzheimer's they are losing much faster. So the problems with the olfactory sense is really one of the earlier symptoms. And this is because we are really accumulating in the olfactory bulb because of the high vascularization. So we are really reaching there, and we are really making an effect. That was really very exciting to see. So with this example, I would like to close the talk saying that I hope that I managed to show that understanding the rational design and the versatility with all the features that are having a role in the design of a polypeptide conjugate, we can really make a difference. And we can really, if we want to really make this difference, we have to think from the very beginning how to make these materials to be able to transfer and to reach the patient. I haven't done anything of this really. It's only my people in the lab. They are really hard workers, interdisciplinary people. I'm really lucky that the team that I have, many collaborators. I'm a person who really loves to collaborate with people that knows much more than we do in the models, in the diseases. So as you see, we have many collaborators, not only in the companies and the academia, also in the clinical scenario, because talking to clinicians is critical to understand the needs for the disease. And for sure, we need the money. And without the funding agencies, we will be able to do anything. So, and thank you so much for everybody too. I'm really happy to take any question from now. Thank you.
Thank you very much, Dr. Maria. A very nice talk. And we have a few questions. And the questions came from all over. Let me see. And the first question is, sorry. The first question is from Hu in Sichuan. And the question is, who says, a very beautiful work. Have you started to develop the platform for mass fabrication, scale up of these nanostructures, or are you collaborating with others to do so?
So depends on which one. For example, as I mentioned, the spin out of the lab is the company who is called Polypeptide Therapeutic Solution, PTS. This is the company we are working with to scale up our careers. So they are already they are a CRO for many other people working with polypeptides, not only us, and they are already doing things in clinical trials that are in clinical trials with also pharma companies, but depending on which ones. For example, those that the cross-link material, for example, is not a scale up yet because of mentioning we need to implement their flow chemistry to go higher to the kilo scale. We arrived to 100 grams, but not to kilo scale yet because we need flow chemistry. But they are doing kilo scale with the Unimes, with the star material, with the linear polymers, not only polyglutamates, with many other polymers with different architectures. So we are working close with them to do that and to implement also in the lab the characterization method and the validation methods to transfer, because we are using prefractionation, LCMS. They are validated techniques that they are using for as a control process points that they need for the scale up.
Okay, very good. The next question is from Jinbo, and it says, thank you for your wonderful talk. I'm wondering, in your in vivo or in vitro test, did you consider anything about their use in personalized medicine? Basically, can you customize them based on?
That's our final aim, really, because it's what we started to. What the way we are approaching this, because we are not looking at the genes itself as most of the personalized medicine is doing, but we are trying the idea for this is having the capability to fine tune the release kinetics of the drug. And also to have, we have a study in the case of the breast cancer I mentioned. So we selected the 4 major subtypes. So what we have done, we have completely characterized each of the subtypes for the commercial cell lines and also for the patients. And what we saw is that, for example, the triple negative breast cancer patients or triple negative breast cancer cell lines, they have a much more reductive environment intracellularly than the luminal A, for example. Mm-hmm. So in a personalized manner, we know that for the triple negative breast cancer drugs that we are using, the linking chemistry that we are implementing are redox trigger, for example. There are other subtypes that they have greater degrees of cathepsin B or cathepsin K, for example, if you go for a metastasis in bones. So understanding the triggers, you can personalize for a specific subtype of patients. That's what we are trying to do with the breast cancer, with separating the subtypes and trying to do these high throughput screening approaches, because this is kind of complex. And also fine tuning, understanding with the patient-derived samples. The idea is to understand the triggers there, to design depending on the triggers and use it in there. That's our idea, and find the biomarkers also.
Very, very, very interesting. But actually, I will follow up on that when we go to the panel, because I was thinking of something completely different when the question came. Now, the last question is about the polyglutamate. How do they compare in terms of treatment of osteoarthritis with injection of stem cells on the damaged cartridge or grafting an in vitro grown cartridge? And then the second part of that is, are these polyglutamates sensitive to hypoxia or mechanical pressure, as in regular bone or cartridge healing?
The thing is, we are not, the work I show you in osteoarthritis, they are not intra-articular injections. This was intravenous administration. So it was really home in there after it was not really affecting for the inoculation. But it's not comparing one to another, is not taking one and bringing the other. For example, in the case of the spinal cord injury, we are combining neural precursor cells together with a polymer conjugate of phosphodil. And what the polymer is doing, for example, is preventing the self-death before engraftment. So we are enriching the cell engraftment because the polymer, is delivering the drug in a sustained manner and making it more effective, makes the cells better, more healthier, and more prolific. We have greater neuroregeneration. So I don't think in nanoconjugates to substitute stem cells, they can really contribute to make it a better therapy. That's what we are doing in a spinal cord, for example. In the case of osteoarthritis, we use it only for diagnostic, and it was IV. So we didn't put the polyglutamates in the pressure in the bone, for example, healing. So I cannot answer this because we haven't done this in the lab yet.
Thank you very much.
Thank you very much. For the hypoxia mechanism. So for example, we have worked with HIF inhibitors. And HIF inhibitors are interesting for us because they are also touching tumor microenvironment. And what we can say is that, and it's something that we are exploring in the lab, trying to understand by cancer metabolomics, the polyglutamates are not a black hole. They are also making a role in a hypoxic areas, but we don't know what yet. So we are trying to understand using cancer metabolomics what is the effect of the polymers in that area.
Okay, very good. All right, so off to, thank you very much. Those are the questions we got from the audience. And we want to go to the panel discussion, and our panelists are going to be Professor Nicholas Purpose from the University of Texas, Austin. I don't think he needs much introduction. Nicholas is a leader in drug delivery. He's very well known worldwide as a pioneer in many areas, published over 1,000 papers with a library of patents and innovations, and has had a lot of awards and accolades from across the world. Our next panelist is going to be Jianting Ding from Shanghai Jiaotong University. And he's a professor of biomedical engineering and also the deputy director of the Institute for Personalized Medicine. And finally, our ex-challenger is going to be Longwei Gao from Southwestern University. She's a graduate student in nanobiomedicine at Southwest University with Professor Shujuan Bao. And with that, we switch to the panel discussion. And the way we're going to do this, the panelists will ask questions, and then we'll give the ex-challenger the first chance to ask questions. So, Longwei, if you have questions, you can go ahead.
Okay. Hi, Professor Maria. Your report is very wonderful, and I have benefited a lot from it. So it's my honor to be the ex-challenger. I have some questions. Firstly, we have designed all kinds of polypeptide-based nanoconjugates, but it's so difficult to achieve the oral administration. So, in view of this problem, how do you think it should be overcome?
Changing the nature of the polypeptide, and there are very nice work done for people like Maria Jose Alonso, for example, in Santiago de Compostela. They are working with polycations, that they are really able to induce this permeation needed for the gut permeation. But for example, the polyglutamates, it will not be an adequate polypeptide to do oral delivery because they will be completely destroyed and not doing anything. But they are polycations with a specific type of coatings and arrangement in different nanoparticles. And there are people with nice work on that area that they are really performing the delivery of different type of small molecules or even peptides or proteins, really. But the nature of the polypeptide should change. So you cannot use a polyanion. You need to use mainly a polycationic system that is really able to disrupt and to enhance the permeation of the gut, or even you can modulate this with molecules, as we are doing, for example, in the intranasal, that you are modifying with different thiol functionality to make it more mucoadhesive, or to derivatizing with lectins, for example, to be more mucodiffusing and mucoadhesive and to enhance this permeation. But it's really, you need to think the way of administration, the design of the system is completely different.
Okay, thank you. And the second question is, in your group, there are so many articles have been reported. So in the future, do you think there's possible to realize coordinated delivery of multiple drugs and the disease treatment in a self-localized manner? So if possible, how to achieve it?
Yeah, I agree with you that this is the way to go. I'm really a fan of combination therapy in that sense. The thing is the control and to know exactly when more things you add in a system, you need to control all the features to be able to transfer, and to the regulatory approval, you need a greater characterization tools. So you need to control the chemistry, and when you are working with aleatory post-polymerization modification, for example, it's difficult to control in which monomer the drug is conjugated. There are methods where the people have started with a drug, for example, Patrick Couvert with the esqualene work has a very beautiful with Nicola, but Julian Nicola, sorry, they are starting with the drug, the polymer, conjugating there, and then making a particle. So there are different ways to control where to put the drugs to make this combination. And you can, and depending if the target you are looking at, or the multiple target you are looking at, are all of them intracellular, or some of them are intracellular, some of them are extracellular, maybe it's better a combo, or maybe it's better to have all of them in the same carrier. So you need to understand where the pathways or the targets that you need to go with each of the drugs to be able to design the adequate combination.
Okay, thank you. And the last question is, could you give some suggestions to the young students and the bachelors about the career planning?
The career planning.
Yeah.
I think you have to really do what you believe, and you have to like what you do. You have to love science, because it's not an easy career. And scientists, we do science because we love science. You know, when I was doing my PhD as a catalyst, I did my PhD in catalysis, organometallic. I was doing dial-Salder reactions as a catalyst for titanium. I was doing titanium thiolates as a catalyst for a dial-Salder reaction. And during my PhD, I'll quantify a lot of enantioselective excess. So I knew, and something in my life changed, and I knew that I wanted to go for more biomedical application. And my PhD supervisors in Spain, they said, you are mad. You are not going to be able to publish. You are not going to be able to come back and get a position in Spain. But so you have to really go what you feel is your goal. So I asked for advice. I found Freshe recommended me to go to Ruth Duncan. So I found Ruth Duncan. I could apply my knowledge on inorganic materials. I'm working with many organic things now. I learned a lot of tissue culture during my postdoc and animal models. And I love polymer therapeutics. And I love to have an applied science. So I know myself, I like this application. basic science is so important also. And there are people that love basic science, but you have to find a way if moving forward is your career, but you have to be passionate for what you do. You have very curious, you have to work a lot. And that's true, because if you want to make a scientist, you have to work a lot, but you have to follow your goals and read a lot. Read also all literature, because there are things that there are a lot of mistakes that has been done. You don't have to repeat them. So I'm telling my students, you have to read a lot. Save time for reading. We don't read too much because there are many bureaucracy to do sometimes, and they're not allowing us to read enough. But you need to read. You need to and to be curious and critical with others also. So be proactive in that sense, and follow, and you will find what you like, I'm sure. So you follow that.
Okay, thank you.
All right, now back to the panel. The panelist, who wants to go first?
Well, I think I can talk first, if I may. Thank you. It's really a nice talk. Actually, I have made quite a lot of notes. I do have like two questions. I think the first one is a very general question. There are a lot of people working on drug delivery all over the world and using different kind of platforms. Sometimes people use nano materials, and sometimes people using exosome or liposome systems. And I know that you have been working with polypeptides. So in your view, what do you compare with different systems? And which one do you believe that has a higher chance eventually to push into the clinical side? Thank you.
I think there is not a general one. I think I believe all of them has their own beauty and their own applications. So you need to understand which is the problem and how you can manage with your technology, because it's true that we are not expert in everything, in everything nanoparticles, but you have to understand the limitations and the advantages of your own technology and understanding the pathological needs. For example, if I work with polyanions or polypeptides with small molecules, I know that if I go to the lungs, for example, for inhalation, liposomes will do better. Chain delivery is much easier with lipid nanoparticles than with polyplexes. But to go for diffusion and to smaller molecules to deep penetrate in the tumor, maybe with a polypeptide, with a polymer conjugate, you can do better. So there is no a beta medicine or a beta nano medicine. So I think you need to fine tune or to find those aspects where your nanotechnology or your type of nanosystem that you have decided could make an input, because you have to understand that the PK/PD is so important and the nature will be very important. Having exosomes or cells as a trafficking has completely different behavior than using. People is going more and more to hybrid system. For example, we published with Bruno de Guest this year a very nice paper on immuno, where we put our polymer conjugate inside a lipid nanoparticle. What you manage to do that with a lipid nanoparticle, you manage to reach faster because they are bigger, you have a better IPR effect. And then with the polymer conjugate, we manage to sustain release and to present better the TLR, the inhibitor that we were delivering. And the polymer has also an important role. So hybrid systems also are important. They are beautiful work with Michelle Bradbury, for example, where they have inorganic particles and polymers looking at diagnostics. There are people working with inorganic, organic material with imaging. And there are many, for example, or we are using hydrogels as a vehicles for intranasal to mucosa delivery, where we embedded the polymer conjugates because we need a residence time that the polymer conjugate doesn't allow us to, and we need to put them in a hydrogel. So you need to understand the way of administration of your nanosystem and which biological barriers, and then you can decide which one is better to do. So I will never say that one nanomedicine is better than another. I think all of them has their own advantages and disadvantages depending on the application you are looking for.
Okay, thank you. I do have another question. Like polypeptide is really very interesting stuff because it consists of a long chain or short chain of amino acid sequences, and as a drug development person, how do you view this? Like, say, eventually when this polypeptide goes into human body, it can eventually be cut down into small peptides, right? Anyway, right? So we know that certain small peptides itself has certain function, by nature. So if a long, peptide gonna eventually be cut into several short peptides, is it possible that some of these short peptides themselves will have a certain, functions, either good or bad? How do you perceive this? How do you avoid such kind of things happening? Thank you.
Yes, I fully agree. That's something that we are exploring because the carriers, the polypeptide carriers, are not a black hole, as I mentioned in the talk. So they are also having an effect. What we have seen, the polypeptides are biodegradable. So for example, Copaxone that is in the multiple, it is already a generic drug. It has a epitope, nobody knows exactly which one it is yet, but it has an epitope that is immune responder. So it activates the immune system and is working for the multiple sclerosis. So depending, one of the things, for example, if you want to use the polypeptides to deliver small drugs to circulate long time in the body, you don't want these polypeptides to be immunogenic. So then you have to check and you have to implement safety controls in your models to be sure that you are not triggering cytokine cascades or any type of hemolytic effect, for example, in your blood circulation. That's in the case of delivering small molecules. And as a control, we are implementing in all the conjugates that are delivering small molecules to a polymer, any site, to a tumor or to the brain or whatever. On the other side, if you want a vaccine, for example, so maybe you want this to be immunogenic, and you want to be immunogenic in the nice way because you are activating the immune system in an antigen-specific manner. So we are looking for carriers and to sequences of peptides that are making this role, and we are trying to embed it. So, and for example, many people is using polylysine. Polylysine per se also helps the immune system to be activated, and there are many things going on with polylysine and different applications because of this immunogenicity. For example, also there are amino acids where specific pathologies are taken up. So for example, the CNS disorders, when you have an ischemia, the ornithine-arginine pathway is very critical. And arginine is an essential amino acid. So you can play with specific amino acids to make your polymer more active and to be even better. In the tumor, the same, the amino acids that can help you to enhance the uptake in tumor cells. And that's something, for example, that is a research lab that I started last year, mainly 1 year ago a bit, looking at the active role of polymer to synergize with drugs, controlling which type of amino acids we are hanging from them. And this is very interesting. And also, if there are pathologies, the cells are kind of a threat. So they are reacting differently in front of polypeptides. That is, if the cells are in a normal state, they are healthy state. So pathological sites react in front of polypeptides in a different manner than the normal sites. So there are very interesting molecular mechanisms going on, and amino acids that are essential, and metabolomics can give you a lot of answers on that respect. So I fully agree with you that this is an important thing to control.
Thank you. Thanks for your answer.
Actually, Maria, if I can follow up on that, because as you are presenting, it came to me that maybe you have an opportunity to also personalize the treatment to have a secondary effect, especially for people who have very rare diseases. For example, people who have a PROS1 gene mutation, where they have a problem with generating certain kind of proteins. Could you design the carrier such that the fragments that are broken down actually help some of those people overcome those deficiencies?
I guess you can, if you understand which triggers, because the polypeptides can be triggered by proteases, but also you can engineer blocks copolymers or brine systems where you can make triggers inside that they can articulate it by pH or by redox that are endogenous triggers, and you can release these fragments. So we have delivered also some peptides or some our proteins itself, and you can deliver for this enzyme replacement therapy. There are things going on that respect, and that could be very useful, the polypeptides that go to lysosome to do that. So for sure, this is possibly done, but you need to understand where this protein has to be replaced, if it is in circulation, if it is in intracellularly, and design those triggers, those bioresponsive sites. It's the same as the cross-linking for a hydrogel, for example. You need to understand which is going to be cleaving and releasing these fragments that has to be active. But for sure, it's possible to do that.
That will be very exciting. So I wish you all the success as you do that. So, all right.
We can talk after the study about this.
Yes, I will be very interested. Professor Peppers, do you have a question, comment?
Dear Professor Vicent, or as we say in Spanish, Vicent, Maria, as we all know you, this was an absolutely incredible talk. It was a tour de force of things that you have done, things that others have done, things that you see going in the future. And the ICANN X talks are supposed to be tutorials, and yours was a great tutorial for the students and for the faculty and for the industrials in the world who listened to it. And I'm pretty sure you had a lot of people from Spain and Portugal and Israel. You mentioned already the collaborations, and the United States. I can assure you many of ours who attended this talk. I have followed something that we follow when we do legal cases on scientific matters in the United States. I write in little post-it questions. So I have about 50 questions. I am not going to answer. No, I'm not going to answer 50. And I would appreciate it if you just give an answer to just one or two questions for the benefit of everybody. One is a personal one, Copaxone. I have always followed what has been happening with Copaxone and multiple sclerosis. I had never understood how a polymer by itself could be a therapeutic agent. And I think you explained it in a magnificent way. It's competing, of course, with Avonex and with other products in the market. But I finally understood how Copaxone works and why it works from what you presented. So that's a comment. That's not a required answer unless you want to give an answer. But the second thing is, I was fascinated in the middle of your talks with the results on cancer treatment. Now, these are preliminary results, and you will be the first one to say, Nicholas, we haven't finished. There are many more things to do. But it occurred to me that they are nanomedicine. They are nanoproducts. And they seem to be working in a very promising way for treatment of certain types of cancer. How do we answer then those who are naysayers, those who say, oh, nanomedicine does not work, it will never treat cancer, it treats only mice, but not humans? I don't know if you want to make a comment or if you want to take a position, but I wonder what you're thinking.
I'm a pro nano person for sure. So I do believe nano has done already an effect, and the best supporting it was I saw from Patrick Coover in the CRS a few years ago. That was a magnificent dance on the pro nano. I believe this bio-nano interface interaction, it makes a major role and a major change of what the nano can do. It's not only releasing in a sustained release, it's the interactions at the nano scale and these biological membranes, what it triggers a different effect. Most of the time, it's hard to understand the final outcome, because sometimes we really spend lots of time understanding why the polymer now is doing this or is triggering this, good or bad. But it's because at the nano scale, it's activating some mechanisms that at the macro scale you cannot see. So I really believe the size and the conformation, it completely matters. We have evidences that the size, the zeta potential, it really can target different cells in the body. It can really trigger different transportations. The work I'm doing with Elena demonstrates that, for example, the lymphotropic delivery, or just changing the compactness, the formability of your nanosystem. It can really make a major role on which cells are you touching. You go into dendritic, or you go into macrophages, or you go in, and this is really changing the output of the final therapy. So I'm a completely pro nano person. The nano scale, it matters, but you need to control in which scale you are to understand.
I am extracting 2 more talks. I wanted to say for our participants and the people who are attending, Professor Vicent was talking about a very famous meeting that took place in Valencia, Spain, 2 years ago, sponsored by the Control Release Society, and she was the general chairman. In that particular meeting, there was a huge debate between 2 professors, one of them, Professor Patrick Couvreur, who will be invited to talk next year in the ICANN-6, and the other one, Professor Keenan Park, my dear colleague from Purdue University, who debated whether nanoparticles and nanotechnology has had any success in the field. That's what she was talking about. Parenthesis also for everybody in the audience, Professor Vicent has been in China. We were together 2.5 years ago in Chengdu. And so when I saw the first talk at Sichuan University, I was very happy because that we were there and we had a magnificent time defining biomaterials. My last 2 questions, one is blood-brain barrier. So Chad Mirking at Northwestern University, excuse me, has come up with some very, very promising results on spherical nucleic acids, and he indicates about 8 to 10% passage through them. And I like these results. There are other investigators who keep talking about they were able to pass through the blood-brain barrier, but they don't really give many data. You talked a little bit about it. Do you think there is a future? Do you think we should expect that someday we will succeed? Because there are certain diseases where we have to pass the blood-brain barrier to treat them, you know.
I hope we can. I think that's my hope, because I believe we need to reach the CNS in an effective manner. With our system, we didn't manage to get more than 2%. So we were not as successful. But it's true that the problem with the BBB is that we still don't understand the mechanism of transportation and the role of the cells that are there. For example, pericytes has an important role because the brain is feeding itself. It transforms many nutrients that are large. So we need to understand this first. So I believe the basic science there and the engineering on the other side merged together could really arrive to something that works nicely. Okay. People is working with gradients of glucose and different physical contents so they can make an effect. But I believe that the future is understanding the mechanism of transportation in the BBB, and for sure understanding this and engineering that respect, we have a future there. But it's a challenging one, but I see that we should still keep working on this area.
Mr. Chairman, I'd like to ask one last question, if I could. First, I want to thank you again, Maria, and I wanted to ask if you could put down this talk in a way that many students and faculty will read it, not necessarily as a review article, but more like a sequence that they will listen to you. Maybe Professor Zhang will tell us that there is already a video. That will be a good beginning. My question is very simple. Do the big companies follow what's happening in polymer therapeutics and try to support it, big companies. Because all of us are familiar, and you are involved with a startup, I'm involved with another startup. Many of us are familiar with startups and smaller companies and so on. And since you said Copaxone, I thought of Teva. Teva started as a small company, and now, of course, it's a huge company. But are the big companies interested, or are they afraid? Because there is always going to be the question, well, what if one of these products fails? Suddenly we have so many litigations, especially in the United States, that we cannot survive.
They are already. I believe they are already. Things are changing. And things people is monitoring, I'm more aware on the polypeptide field. There are a lot of companies, small biotechs, but also large pharma, that are starting to explore. They are not telling you all because they are, but they are really interested on what's going on polymer therapeutics, in nanosystems, and there are quite a few doing things. I don't know how much they will, how long they will take to have a product in there as a new one, but for sure I can tell you that there are big companies exploring polymer therapeutics for very different applications, from going from gene delivery, from from regeneration. So I think they are less afraid now than they was before, because they understood better the problem of the field of polymer therapeutics and many others is that the path has been very tedious, and now we have a better frame and understood better the steps that we need to follow to transfer and to go there. It's true that there are complex mechanisms, but also they are very conservative on that side. So I believe they will be really first like more conservative. When this is safe, they will go to the next step. So this is, I believe they are already in there, and I hope this exponential growth will be maintained, and polymer therapeutics, and if polymer drug conjugates, that is only a small one there, get better.
Thank you.
Thank you very much. For the interest of time, I want to say let's continue this conversation on February 25th next year, when Chad Mackin will be on this stage talking about very similar topic. Very interesting talk. Maria, if we're in person, I believe Alice and I would be walking across the stage and handing you this beautiful appreciation certificate, but now we're going to have to do it virtually. And then thanks everyone for participating. It's a really wonderful and important topic.
Thank you very much. A huge pleasure from my side. Thank you so much for the opportunity to share my work.
Thank you. And then I want to remind everybody else that next week we are going to have Mark Allen from University of Pennsylvania. He'll be hosted by Alice and Shinyu from Toronto, and these are going to be our panelists. And with that, I want to say thank you very much. Thank everyone for attending.